Well, this is kind of an improvised video, but I wanted to show you first of all two things, which is that this guy right here, if we go to his comments, right here, we go to most recent, he's challenging me to make a video debunking him, even though I have already debunked him using just the comment section of his video. You can read a detailed debunking of what we have been discussing right here. But also, if we go to his community section and go to his article and expand it, because the images are reduced for some strange reason, we can see that oxidative stress, which is induced by reactive oxygen species, has an influence in stimulating anabolic signaling, which is muscle growth, which is myofibrillar growth, because ROS can act with a signaling molecule to activate the mammalian target of rampacine, which is the main target by which satellite cells are activated through IGF-1, which is required for myofibrillar hypertrophy, and MAPK, which is also something that is related to myofibrillar hypertrophy pathways. If we go here, it is even more funny because lactate is a metabolic waste product that is mainly produced in aerobic training or high reps, high volume training. And in the same study in which he is talking about, you can see that it says that lactate is one of the metabolic products of glycolysis, and it is widely accepted as an important energy source for many cell types because your cells reutilize lactate to produce ATP and therefore facilitate muscle contraction. If you go right here, it says satellite cells, which are adult skeletal muscle stem cells, are the main players of the skeletal muscle regeneration process. That's right. But not the only way by which you can increase myofibrillar size. We have already debunked all of that arguments in the last video. Recent studies have proposed a metabolic switch to increase glycolysis in activated satellite cells. Moreover, lactate has been shown to affect satellite cells and myoblasts in vivo and in vitro. Myoblasts are the things in which satellite cells differentiate. So lactate has a relationship with the differentiation of satellite cells, which even isn't really regulated by external substances, but it is moreover regulated by the cell cycle of the satellite cells that they enter after exiting the quiescent state, because in each part of the cell cycle different genes like MYOD, MYF5, we have already reviewed all of that in the last video, activate subsequently and signal each of the events that occur in satellite cell proliferation, differentiation and fusion. So it's internally regulated. But even so, these satellite cells have to stop being quiescent to fuse to the myofibers, right? But well, right here it says that lactate has been shown to affect satellite cells and myoblasts in vivo and in vitro. So it really activates satellite cells. It's something that constantly changes their fate, and also that makes them proliferate and therefore fuse to the muscles. During homeostasis, satellite cells exit in a quiescent state, okay? Upon injury, but not only upon injury, as we have already proven, satellite cells enter the cell cycle to become activated and undergo several runs of proliferation, a process that happens very quickly from the first days after injury. Doesn't require an injury, we have already proven that in the last videos, and any substance that causes satellite cells activation will cause fusion, because that is internally regulated by the cascade of genes that activate each time a satellite cell activates. But the thing is that it is really funny that the study that he has posted in his community section is completely debunking what he says. If we go to lactate, not only because of the oxidative damage section that proves that oxidative damage is something regulating satellite cell fusion, proliferation, and activation, because it regulates the path by which these three processes are regulated, which is the mTOR path and the IGF-1 signaling pathway, right? But if we go right here and we search for lactate, we see that it is a signaling molecule affecting satellite cells' fate. And if we go right here, we are going to find in this figure right here different mechanisms in which metabolism can affect satellite cell fate. Satellite cell activation is associated with an increase in pyruvate oxidation because this is a metabolic waste product that you produce each time you exercise, right? This increase generates more citrates production, which is a chemical that appears in a certain part of the Krebs cycle, which is right here, linked to more acetyl CoA for histone acetylation. This is a very difficult process because this is the Krebs cycle, which is this one, which has a lot of steps and is really difficult to understand, but well, that doesn't have anything to do with that. And well, activated satellite cells increase pyruvate oxidation, and therefore this results in higher histone acetylation and an activated myogenic transcriptional program. What does this mean? That myogenic transcriptional factors, which are mainly genes inside of the cell, that is what a transcriptional factor is. Well, it really is a RNA messenger, which is created by copying a certain gene inside of the of the nuclei of the cells. But well, the thing is that activated satellite cells increase these myogenic transcriptional factors that well regulates all of their processes, right? It is not regulated by external things, as many people think. Only the differentiation in myotubes, in case that an entire portion of the myofibers has been broken or the myofiber itself has completely died. Only in that cases differentiation occurs, yes, because they have to create the same myofiber that was destroyed before and replace it, which is not hyperplasia because we are not producing additional units. We are just replacing those that had been broken partially or completely destroyed. Okay, so apart from that, in the hypertrophy process, in which satellite cells differentiated into myoblasts, not into myotubes, because myotube differentiation requires other factors that are regulated in the inflammatory process, like macrophages. The macrophages regulate differentiation, also IGF-1, but that's secondary. In the case of satellite cells that are going to just fuse to myofibers to increase the myonuclear size domain, yeah, they do not have to go through differentiation, okay, and all of the processes previous to that fusion are regulated inside of the cells, yes, by their own genes that activate in certain sequences. If we go to my docs right here and we go to this page right here, we can see that just exactly when satellite cells are activated, a cascade of genes are regulated because of the own cell cycle of the cell, okay, in a certain order. This is a process that starts and ends each time a satellite cell, which is going to be committed to growth, because asymmetrical division produces two satellite cells that are going to have different functions, but this is produced every single time that satellite cells are activated. This this asymmetrical division because you have to maintain the satellite cell pool, so some of the satellite cells have to go back to quiescence to keep the pool big, and in each asymmetrical division, which happens each time satellite cells proliferate, one cell which will go back to quiescence is produced, and another cell that is going to be committed to myofiber fusion or creation of myotubes is produced. Okay, that cell which is committed and is going to be creating a myotube or directly fusing into myofibers, which are two different processes that we have explained in the last video, which are regeneration and hypertrophy, which are not the same processes. Okay, the first one of these processes, hypertrophy, is internally regulated by the satellite cell. No extraneous factor regulates it. Each time a satellite cell activates, which means that it starts its own cell cycle, these genes right here, PAX7 and YF5, will activate in succession, okay, to lead to satellite cell fusion. So it doesn't matter if it was activated by damage or anything else, they will fuse, because this is internally regulated, not externally regulated, which means that these substances that damage releases, that microinjuries release, do not affect where satellite cells will fuse or not, because that is internally regulated. Okay, but going back to this, it basically says that lactate may have some role in the metabolic control of satellite cell fate with a transcriptional factor inactivation, favoring satellite cell activation. You're basically saying that lactate right here makes satellite cells activate, proliferate, and therefore fuse, because that's internally regulated. The mitogens released by damage don't regulate that at all, because that, as I have already said like 5 times, is internally regulated. And well, you have just proven you wrong again. You're a complete clown. You have proven you wrong in the in this study, in the 2 things that you constantly claim to not be related to myofibrillar growth. So that's really sad. And let's also see the video that you have posted right here, trying to explore this article to see
All right,
what
friends,
you
it's
say.
I'm back with another video, and I posted this in my community section. I just did a video talking about some, those drug addicts again, steroid drug addict people. But this has nothing to do with them. This has to do with the driver of hypertrophy. What is the main driver of hypertrophy? It's really simple. You want to know the main driver of hypertrophy? And they tell it to you just about in every one of these reports I brought out before. What does it say here? Skeletal muscle hypertrophy depends on what? Positive muscle protein balance. Protein synthesis exceeds breakdown. Thus,
Yes, but protein synthesis exceeding protein breakdown is not enough. If you watch my previous video, I made an analogy in regards to a house and bricks. Just having enough bricks for building a house, or well, having more bricks than what you're breaking, does not mean that you're going to have a house. You need builders and also a stimuli to want to have a house, because you can have all the bricks you want, but if you don't want to construct a house, you are not going to do so. So this is not enough to build muscles.
is excellent for the stimulation of an anabolic signaling and promoting of muscle hypertrophy. So what is good? There's all kinds of damages. Okay, I got crazy people that are writing me all the time in my comment section. You get these nutcases that come back and they're trying to tell me what I'm doing or what I'm supposed to be doing or this or that or something. I know what I'm doing. You need to know what you're doing. So anyways, the point is that is the major driver of hypertrophy. Hypertrophy just means something is getting bigger. Now, what is the driver of muscle cells?
It is not the driver of hypertrophy. It is needed for hypertrophy. You have to distinguish between 2 things: a cause and a necessary antecedent. Muscle protein balance is necessary for building muscle, but it is not the cause, because it is not enough to build muscles. You can have all the protein you want. If you don't have a stimuli for it to go somewhere, it won't go anywhere.
What is it? Okay, here's the thing. If you get more muscle cells, if you damage muscle fibers and the progenitor splits, divides, and increases in number and binds with the cracks and the domes, not the cell swelling, the cracks and the domes, not the swelling of
when did I say that it is going to fuse with the cell swelling? First of all, satellite cells don't only fuse to cracks and domes. Has been completely debunked in my last videos. But also, satellite cells, once they have been activated, they will fuse with the myofibers they enter in contact with. Because in order for them to donate their nuclei, they have just to be in proximity with the myofibers. So if they have been already activated, which means that they will proliferate and migrate, and well, these processes, the first one of these processes, the proliferation and activation, are regulated by internal factors, but also the migration is regulated by, in this case, external factors like chemoattractants that are released without damage, like NO. In this case, you can see this example right here, which is a substance that is released each time your muscle stretches or is damaged. We are talking about the two things as one independent to the other, stretch or damage. If we were only talking about damage, and therefore stretch-induced damage, we would only say damage right here, okay? Well, the thing is that once satellite cells are activated, they will fuse. Cell swelling activates muscle satellite cells, as it was explained in your comments exactly by this cycle right here, which you can easily see if you go here to the 12th citation right here. You put it on Google, you go to this article right here and you read it. You will see that it ends up causing muscle protein accretion, which is a thing that causes myofibrillar growth. So cell swelling causes satellite cell activation because it causes muscle protein accretion, and it initiates anabolic and anti-catabolic processes. So it regulates muscle satellite cells. Once they are activated, okay, these satellite cells, once they enter in proximity with a myofiber, they will fuse, okay? What you're saying right here has no meaning whatsoever. Let's repeat it once again.
The progenitor splits, divides, and increases in number and binds with the cracks and the domes, not the cell swelling. The cracks and...
No. The cell swelling, first of all, is not an object. You are committing a refutation fallacy, so it cannot fuse to the cell swelling. It is a state in which cells can be. It is not a thing, therefore it cannot fuse to that. And we have already debunked the fact that they can only fuse to crack and domes. Satellite cells only have to be in proximity of myofibers to fuse with them, so that's false, simply false. No evidence was provided whatsoever that explicited why you require that only crack and domes can that only crack and domes permit satellite cell fusion. You provided no evidence to show that satellite cells will only fuse in case of the myofibers having a crack and a dome in a certain place, which is also false considering that the same amount of satellite cells are being activated even if the myofiber only has damage in one of its zones, in only one of the places along all of its longitude, which has been shown in the last video. So this already proves that satellite cells will not only fuse with the injury, because there are the same amount of satellite cells and less damage, and therefore less zones with a crack and domes for them to fuse into, so they will end up increasing the size of the myofiber, fusing to zones which are not crack and domes. Yeah, because there are less zones and there is the same amount of satellite cells, so they will have to fuse to a myofiber even if there are no cracks and domes in that places. But also they only have to be in proximity to myofiber to fuse with it. Okay, that thing about crack and domes being the only places to which they can bind to comes completely out of nowhere. It can be the case that they fuse to that places, okay, in the regeneration process, not the hypertrophic process, but it doesn't mean that it is the only place in which, to which they can fuse. So that is false.
The dose, not the swelling of a cell. You understand me? And it binds there. Yeah? Okay. So you get more muscle cells. Is that hypertrophy? Yes. Can you see it visually? No. You have to see it under a microscope. Now, to see those cells, hypertrophy, to get them really big, to see that mass, to see that mass, protein synthesis needs to what? Exceed that breakdown to see that mass.
That's a necessary antecedent, not a cause. That is not the stimuli that you need for that protein to be used, so it's not enough.
And not under a microscope. Get it, bro? Yeah. Now, as far as cell swelling, I talked about this. That's what's in this report here. They're talking about the role of metabolic stress. In a metabolic stress, it's a cell swelling. You're swelling the cells. Your cell swelling, like it says here. It's through hypoxia and ROS. Hypoxia. Yeah, hypoxia and ROS. It's swelling the cell. You're a chronic lifter in the gym and you're lifting. So your metabolic thing has changed. It's not based. Your lifting is not based on muscle damage. It's not based on these cracks in the domes.
ROS, which is oxidative induced damage, because a reactive oxygen species cause oxidative induced damage, cause growth. Because the thing that makes myofibers receiving injuries and growing after that are the mitogens that are released by the structure of the myofibers being compromised, okay? When they receive micro injuries, the insides of the myofibers go to the outside and reach the satellite cells to activate them. That is the mechanism by which micro injuries cause satellite cell activation. Guess what? Reactive oxygen species also cause oxidative damage, which is a form of damage that can compromise the structure of myofibers, okay? If the structure of myofibers can be compromised by not only ROS, but also osmotic pressure, as it is explained in the other comment that I made you, that we are going to bring up right now, then they both are a cause of damage, okay, first of all, and a regulator of satellite cell fusion and activation. It is said right here in the comments that you didn't even show,
All right.
because you are really scared to directly reply to my comments, and you have to make a video that does not mention me. You go right here, and it is explained right here. Okay? It does not make sense to say that swollen cells do not promote myofibrillar growth, not only because osmosis compromises the structure of cells via shear forces. Also, we have this thing right here that explicits the fact that it causes initiation of the anabolic and anti-catabolic process, which is a state that you need to grow, and also muscle protein accretion, exercise-induced cell swelling, we're talking about that, but also not only because osmosis compromises the structure of cells via shear forces that stretch it and cause the release of mitogens, because compromising the structure of the muscle cells, or even compromising the external cellular matrix, which is something external to the myofibers, cause the release of mitogens, because mitogens are released when the structure of the muscle cells it receives a stress. Okay, not every stress breaks you, right? Not every stress breaks the structure of the myofibers, but stress is needed for myofibers. For the structure of myofibers to be altered and for mitogens to be released. Even so, sarcomeric sensors do not work by mitogens, and they also activate satellite cells, but that's another thing. I'm completely lost right now. Let's read it again. Not only because osmosis compromises the structure of cells via shear forces that stretch it and cause the release of mitogens that are the same things that trigger satellite cells in the case of microinjury and cause their fusion, but also because of osmosensors, a type of mechanosensors. You have mechanosensors all around your body, not only in myofibers. When you touch something and you feel the touch itself, that's a mechanosensor, a type of mechanosensor. You also have sarcomeric mechanosensors, which send signals to the satellite cells to activate, proliferate, and fuse after they detect a stretch just a stretch. They do not care at all about damage. They do not detect damage. They do not have any mechanism to detect damage. Just a stretch. They send a signal for satellite cells to proliferate and fuse. That's the way they work, and it also has it also makes a sense when you contrast it to human evolution, because you would also obviously develop a mechanism by which you can grow without constantly making yourself prone to damage. Okay, it doesn't make sense when you think about evolution. So it's obvious that the human being would develop a mechanism by which its muscles can grow without himself suffering damage. Okay, so it makes sense for mechanosensors to be present also when you talk about evolution. Okay, also because of osmosensors present in the fibers whose specific job is to detect that the cell is receiving a stretch under load. Okay, so that already debunks what he's saying, but let's go.
It's not based on muscle injuries, it's based on cell swelling. You're swelling the cell, so you're getting inflammations through cell swelling. Every time you come to...
Sorry, inflammation has nothing to do with cell swelling. Swelling means that the amount of liquid that is inside of the cells is disproportionate to the size of the cells, and it causes the structure of the cell to stretch. Okay, it has nothing to do with inflammation, even though you need it for growing, because the inflammatory process, if you watch this thing right here, follow acute injury and the changes in gene expression in muscle following injury. These inflammatory cells and this inflammatory response occurs each time you damage your muscles or you require inflammation to grow, and also the macrophages that are released during the inflammation process is needed for myotubes to be produced and replace those myofibers that have been completely broken or have lost an entire segment. So that is also wrong. You also have this section right here talking about inflammation and also debunking you, but you obviously aren't going to read it because you are scared of something that debunks you. But let's go.
To the gym, you're swelling the cell and you're hypertrophying. Get it? Yeah. Now, if you want that cell to grow mass on it through like proteins, well, the protein synthesis needs to exceed that breakdown.
No, cell swelling is temporary. Okay, if cell swelling were to be the cause of your muscles being bigger, then you wouldn't grow across the long term, because this would be just transient hypertrophy. Okay, so if you receive muscle hypertrophy from cell swelling, which means steady growth throughout a certain period of time, which was already proven in this study, but okay, if you constantly grow by stimulating cell swelling, you are not growing because of the cells being swelled with liquid, because that only happens at one moment in the day and reverses to its original state after training. If your muscles normally, okay, are bigger in normal conditions without having trained or received any kind of pump, we are talking about a long-term hypertrophy, which cannot be explained by this transient hypertrophy, because you cannot accumulate this kind of hypertrophy. Okay, each time you get this hypertrophy, you are going to lose it. Each time you get the pump, you lose it. You get the pump, you lose it. So you cannot accumulate growth. So if after a year of stimulating cell swelling, you have seen some growth in your muscles, the same thing applies to glycogen loading, by the way. But if you want to see a complete debunking of glycogen loading, watch the other video. Okay, so if you see a steady growth by simulating this mechanism, which is cell swelling, you cannot explain it by the cells being swelled, because this swelling cannot accumulate over long periods of time, cannot produce accumulative hypertrophy. Your muscle cells cannot accumulate swelling. Okay, each time they swell, they are going to stop swelling after a certain period of time. So if you grew, if you have grown, it means that it has stimulated growth apart from just the visual aspect of cells being bigger. And also, cell swelling occurs at a microscopical level. Okay, so you cannot detect a increase in size of the muscles just by swelling of the cells inside of them, because the swelling inside of the swelling of the muscles inside of them, the swelling of the cells. Sorry, I am completely missing the words that I want to say. The swelling of the cells inside of your muscles, okay, cannot be noticed by the naked eye because it is really microscopical. So if you notice growth, you cannot just explain it by the cells being swelled. Okay, that doesn't make any sense.
But none of you are doing that in the gym. I try to explain this. None of you are eating in an anabolic growth state to suppress, to exceed that protein break.
It seems that he knows all the 32 of the viewers that are watching him, I suppose, because he's saying that none of them are doing that. But okay, let's keep going.
Down. Because proteins are being broken down all the time. Get it? Like it says here. You want me to read the whole thing?
Not all the time. Yes, you are constantly receiving oxidative damage, and also that's one of the positive effects of exercise. It makes oxidative damage less, okay? So since the amount of damage that you get from the constant oxidative stress in your day-to-day life is less, you are going to live more because of that. This another one of the positive effects of the repeated bouts effect. We have already mentioned them all in my previous videos. I won't go over it again, but you already get it.
For your resistance training, skeletal muscle hypertrophy depends on what? Positive muscle protein balance. Protein synthesis exceeds breakdown.
Yes, depends on is not the same as that being caused by the thing you're mentioning. We have already debunked this. I won't go over it again.
Thus, RT is excellent for stimulation of an anabolic signaling. Yeah, signal, that's all. And the promotion of muscle hypertrophy, the promotion.
Yes, anabolic signaling is exactly increasing the amount of protein synthesis and increasing the myoprotein accretion of your myofibers, which means growth of your myofibers. So if it signals growth of your myofibers, you cannot just say that that it's something insignificant, because it's something that you really preach. So that was really strange coming from you.
But not hypertrophy. Metabolic stress is one of the primary mechanisms that makes RT increase muscle mass. Yeah, metabolic stress is one of the primary mechanisms that makes RT increase muscle mass. But without protein synthesis, it won't increase its mass. It's also increasing its...
Yes, you have to have a positive protein balance to build muscle, but it is not the cause.
It's swelling, so it's increasing its mass. Get it?
Yes, but swelling is not detectable by the naked eye, so you cannot explain that your muscles have grown because of the swelling of the muscle cells. And also, you cannot explain that they have grown in a period of 1 year because each time you swell, you are going to lose said swelling. So, along a period of 1 year, you should have grown nothing at all, okay? The same goes for glycogen loading. Each time you glycogen load, you are going to, after a certain period of time, lose all of the glycogen that you have loaded because of it being used by exercise, or it being completely, how can I say it? I don't really know the term right now, but it being no longer useful, and therefore it being just taken outside of your own body. Okay, that's the way in which you can manage glycogen, because your muscles don't need it anymore, and where your body gets rid of it. But both of these types are transient hypertrophy and cannot explain growth in 1 year. Okay, so if you have grown, you cannot say, oh, those are cell-swelled muscles or sarcoplasmic muscles or glycogen-loaded muscles, because glycogen loading, each time it occurs, it will stop occurring. So you can't make a cumulative process along 1 year. You are going to stay the same, literally the size of your muscles will be the same, at day 1 of the year and day 365 of the year. You will remain the same. If you have detected some changes, you cannot explain it by those two mechanisms mentioned before, because of the reasons that I have already mentioned.
Temporarily, mainly due to the rise of what? Anabolic hormone release, hypoxia, ROS production, and cell swelling. Does that sound to you like muscle injury, like muscle damage? Does that?
Yes, ROS is reactive oxygen species, and what do they cause? Oxidative damage. So you're completely stupid. And also hypoxia creates an environment in which muscle cells are not receiving enough blood. Okay, that's hypoxia, and it obviously is going to cause necrosis and damage of the muscle fibers. You don't have any single ounce of common sense inside of your brain.
That sounds like micro injuries to the fiber.
Yes, and they also trigger the same mitogens that produce the cell activation because of the production of micro injuries inside of your muscle. So they work by the same mechanism by which micro injuries work.
Listen to what they say here. Hormonal release, hypoxia, ROS production, and cell swelling. What does that have to do with?
Yes, what does it say? What does it say? And also something very funny is that he said that metabolic stress is important for muscle mass, and now that he's talking about all of the things that metabolic stress regulates in order to increase muscle mass, he's trying to criticize it. He's literally contradicting himself in a span of less than 2 minutes, so that's very funny. But it says it right here, okay? Metabolic stress is one of the primary mechanisms that makes RT increase, which is resistance training, increase muscle mass, mainly due to the rise of anabolic hormone release, which is needed for your muscles to grow, and is the reason why men grow more than women, because of the increased amount of anabolic hormones. So it is really important that you have a good amount of anabolic hormones being active in your blood. So that's something positive that we take out of metabolic stress, and that impacts myofibrillar growth. So that's something that really goes against your principles, because metabolic stress is mainly caused by the pump and by high reps and high volumes. So it completely debunks what you say about supposedly chronic lifting, which is kind of funny. But let's see what he has said before,
Micro injury.
And let's debunk him again.
The fiber. Listen to what they say here. Hormonal release, hypoxia, ROS production, and cell swelling. What does that have to do with micro injuries? It doesn't.
Well, I have already explained why it has to do with muscle damage, and even though specifically cell swelling does not trigger satellite cell activation and fusion by muscle damage, but it can trigger it by muscle damage, because the cells can swell so much that internal factors are released to the outside because of the cell structure of the cells breaking, but also not only because of osmotic sensors that I have already mentioned that play a role in increase of myofibrillar protein aggression and satellite cell activation, but also because of a simple mechanism, which is solute gradient. Okay? When a cell has a lot of water inside of it, when you have a cell with a lot of water inside of it, the things that are inside of the cell are going to be spewed out to maintain the overall balance of solvent and solute, okay, concentration. And that's why mitogens are going to be released to the outside and are going to activate satellite cells by cell swelling, which is going to cause myofibrillar growth. So not only it causes myofibrillar growth by damage, but it also causes myofibrillar growth without damage, okay?
You understand me? Even if the progenitor is activated here, which it will activate for sure. It will activate in there. It's going to activate, definitely. And I posted another, lactate metabolism and satellite cell fate. During homeostasis, SCs, the satellite cells, are in a quiescent state. We know that. I've done this in over 500, 600 videos already. And I've done it on my old channel even more, like so many times. Low metabolic demands. Yes, low metabolic demands. Okay? That's when it's quiescent. But under stress, however, caused by injury,
Injury, exercise, two different things. They are talking about the two things like completely separated by one to the other. Because if they were talking about exercise-induced micro injuries, they would only say injuries because it already encompasses that category. So this article right here is saying that you don't need damage to grow, just exercise. It also debunks what you say again.
Size or other variables. SEs become what? Activated to proliferate, differentiate, and fuse to regenerate the tissue. But sometimes it just proliferates. It doesn't differentiate, and it doesn't.
Sometimes it just proliferates. Let's see from where has he gotten that fact. We don't really know. He hasn't posted any evidence from that being the case, and also is false because of the mechanism by which muscle satellite cells regulate all of these processes, which is internally, which means that each time they activate, they are going to do that because they are programmed to do that at a certain time. It is regulated by genes inside of the satellite cells, which are upregulated each time a certain amount of time has passed, okay? Because they are part of the cell cycle of the satellite cells. You can see it right here. So what you're saying is complete nonsense. Not only haven't you provided any proof for what you're saying, but also because this proliferation, this activation, and this differentiation is going to have to happen anytime satellite cells are activated. Also, what are you even thinking about? Do you think that satellite cells just activate and you keep them around your muscles doing absolutely nothing? It wouldn't even make sense from an evolution standpoint, okay? But
doesn't fuse to regenerate tissue. Depending, if that tissue has been damaged, then it's going to have to regenerate that tissue. That doesn't mean you're growing a muscle. You understand me? That doesn't. Look, a cell is swelling due to hypoxia and ROS. Okay, and what about it? What about it? Okay, let's say the ROS.
Yes, both of these mechanisms cause satellite cell activation and fusion, and they also cause damage, which is a driver of satellite cell activation and fusion. So, what are you saying?
Doesn't look, a cell is swelling due to hypoxia and ROS. Okay, and what about it? What about it? Okay, let's say the ROS activates the satellite cell, and what? Is the cell damaged in any way? No, it's swelling. It's swollen now. It's swollen. So what's going to do?
Yes, but it is going to stop swelling, and also satellite cells can fuse to swelled cells. Nothing stops you from doing that, because the swelled cells we're talking about are myofibers, okay? Satellite cells just have to come into contact with the myofibers, which is the mechanism by which they donate their nuclei, and they will donate their nuclei. They do not need to be damaged. That's all. It's really simple. I have already explained why they don't even need to be damaged. They don't even need these cracks and domes. And let's see if I can find the article that was talking about that, which is migration. Let's see. Where is it? Edit. Migration. Again, migration. Where is it? Cannot find it right now. Let's search for the word migrate. Yeah, right here. This article right here. This result suggests that mitotic activity of satellite cells after localized injury occurs mainly at or near the site of injury, but that many of the dividing cells have migrated from undamaged areas from the distant from the lesion site. So even if you receive less damage to that myofiber, the same exact amount of satellite cells will activate, and since they are more there are more satellite cells than what you need to seal that micro injury, they will fuse with other parts of the myofiber, and they don't have any impediment to do so because they only have to come into physical contact with the myofiber to donate their nuclei. That's how satellite cell fusion works.
Do nothing. It's simply proliferating because of hypoxia damage and ROS damage. It's damage to some tissues in the body.
Yes, if they are proliferating, they are going to fuse because it is internally regulated, okay? It is not regulated by any single thing that is only released when muscle cells are damaged, okay? It is internally regulated. It doesn't matter by which method you have activated the muscle cells, because the method by which you activate the muscle cells only, the muscle satellite cells, only changes, okay, the substances that are released to the outside. And the muscle satellite cells don't really care about the substances that are really being released to the outside for the purposes of fusing to the myofibers. They don't care at all. They don't care at all about that, because that's internally regulated by their own cell cycle, which upregulates certain genes for them to proliferate and fuse and also differentiate. And you don't even need differentiation into myotubes, which is what the stage of differentiation is referencing to, for them to fuse to myofibers.
Body. So it's just moving, or it's proliferating there, searching, lurking. You activated it now. You're exhausting the satellite cells now. You're not split, dividing, increasing in number, them going back for future regenerative demands and increasing in number.
What are you even talking about?
You're exhausting the satellite cells now. You're not split, dividing, increasing in number.
You're not split dividing the satellite cells. Each time satellite cells are activated, you increase the pool of satellite cells, because the asymmetrical division creates quiescent satellite cells and increases the count of them. Each time you activate satellite cells, the symmetrical division happens. You have it right here in this article, and you can read all you want. It happens any single time satellite cells are proliferating, so that doesn't make any sense.
Number of them going back for future regenerative demands and increasing in number. You understand me?
Yes, they are going to increase their numbers, because each time the satellite cell pool is depleted, which is the case that you have described, the amount of satellite cells that you're going to have for the next time you use them is going to be greater. That is the mechanism by which that adaptation occurs, by using your satellite cells.
Through micro injuries, because your muscles, they don't micro injury all the time, man. Get it? They're protected.
Yes, but you don't need them to micro injury, and also if you don't want the repeated bout effect, have luck with not training for 8 to 12 weeks, okay? Because that's the amount of time that you have to wait right here, 8 to 12 weeks. You can read it. Newly regenerated fibers become susceptible again to eccentric exercise in 8 to 12 weeks. But also you want to remain with weak muscle fibers that can completely break with a minimal amount of force exerted, so that's dumb. And also I have debunked periodic lifting in the last video, so let's go.
It's a highly protected state, okay? It's under many protections. It just doesn't damage just like that.
Yes, because you are not supposed to completely destroy your muscles, okay? Because that doesn't even make sense from an evolution standpoint. Why would you want to make the only mechanism by which you can grow damage or destructive damage that completely breaks your muscle fibers? Not to mention that the repeated bout effect is necessary for damage, for growth. We have already proven it in the last videos, and we have this study right here. You can read if you want. It shows that you need a repeated bout effect to grow.
I'm gonna wave my arms up and down and go to the gym, and they're gonna get damaged, and I'm just gonna get bigger magically on a breatharian diet. Man, people are really insane out there. Okay, transcriptional cascade bar, whatever. They talk about a whole bunch of other things here. Okay, they even talk about antioxidants can modify protein signaling after RT sessions and impairs muscle gains. So antioxidants can impair muscle gains. There you go.
Yes, because you have just proven. No, this is completely stupid. I cannot even start taking you seriously. You have just proven that you need oxidative damage to grow, because if using antioxidants decrease the oxidative damage, and while this is happening, the amount of growth that you receive is less, then there is a role for reactive oxygen species, which are ROS, for myofibrillar growth. You have just proven you wrong with the thing you read. You're completely dumb.
Be fucked after your session. It's widely accepted as an important energy source for many cell types. This lactate bullshit, whatever, metabolic products. Propose actively participate in cell-to-cell communication. Satellite cells, which are adult skeletal stem cells, are the main players of the skeletal muscle regeneration process. Yeah, it's a regeneration. It regenerates muscles, yes.
No, they do not only regenerate your muscles. It's not the only role that they have. Satellite cells also fuse with your muscles to make them stronger, and the regeneration process has nothing to do with the hypertrophic process. You can see it right here. They are functionally different. One of those only cares about replacing a myotube that was, a myofiber that was severely damaged, and the other one only cares about growing your myofibers, even if they have received damage or not. It is explained right here.
Recent studies have proposed a metabolic switch to increase glycolysis in activated SCs. Moreover, lactate has been shown to affect SCs and myoblasts in vivo. In vivo means it's been a laboratory, but whatever. In vivo.
No, in vivo does not mean in a laboratory. It means inside of the being that you are analyzing. In vitro means that it is being analyzed in a laboratory. You have completely mixed up the terms.
And in vivo, and in whatever, vitro, and in vitro, vivo and in vitro. In this short review, we describe how metabolic variations relate with SC fate, quiescence, activation, proliferation, migration, differentiation, fusion, and self-renewal, as well as discussing possible relationship between lactate,
Okay, so now lactate, which is a metabolic waste product that is released when you receive a pump and you go into high reps and high volumes, now regulates all of that, which even though you have said the exact opposite some minutes ago.
And so, yeah, there, they tell you again, okay, it's in a quiescent state. Upon injury, SCs enter the cell cycle to become activated and undergo several...
Yes, but not only upon injury. I have already debunked this so many times that I don't even want to go at it again. You can see my previous video, and even in this video I have talked about it.
Several rounds of proliferation and process that happens very quickly from the first day after injury. During proliferation, systematic and symmetric.
Asymmetric, not systematic.
Divisions happen, yeah. Symmetrically,
you haven't even read the article that you're criticizing. That's very funny.
Planar divisions can give rise to two activated SCs or two SCs concomitant to myogenesis, whilst asymmetrically divided cells.
You can already see that he isn't even understanding what it says. He's already completely lost when it comes to this article. He's reading everything really fast and not comprehending any of it, and then he goes and makes these kind of videos, which are incredibly lame.
Apical basal divisions. We give rise to one activated SC and one quiescent SC, which contributes to self-renewal SCs. That's my training that I do now. I do regeneration training because I know exactly what I'm going to get. I know what I'm looking for. I know how to get them. I know all about.
You don't know anything about that, and you have proven not only this with this video, but also in the videos that I have debunked in the past. So funny.
Swelling, ROS, hypoxia, cachaxia. I know all these things that cause different damages to muscle fibers.
What are you even talking about, cachaxia? What the fuck was that?
I know what I'm going to get. I know how much I'm going to get. I know what I'm going to shoot for. I know how to be in an anabolic.
You do not know any of those things, providing that you don't even read the articles that you post in your community section.
Growth state. I know how to use complex carbohydrates to create more of this sarcoplasmic and even make space for more myofibrillar.
No, it does not have anything to do with creating more sarcoplasmic. It has to do with glycogen loading, which has nothing to do with sarcoplasmic muscles. I'm not going to go through it again, but well, you can see my last video inside of my channel addressing exactly that argument. So it is a conclusion from this video. If you want to debunk the argument, watch this video and debunk it.
Myofibrillar growth. By doing that, I know that. Shit, man. I know, just whatever, bro. I know how to be an anabolic growth. I know how to build muscles, okay? I know how these satellite cells, when they're activated, I know under what the most activation, the most damage, not getting damage, fucking eccentric, concentric movements, what they do, what they're supposed to do, hormonally, strength-wise, damage, this kind of damage, that kind of damage, chronically lifting, fucking cell swelling in the gym, like a yo-yo, like Blaha, and all these fucking people, bro, get it? It's complicated. I have to make a 10-hour long video, bringing out every report, trying to explain all these things. That's why I do them piece by piece, bit by bit in videos. And then I get some crazy person in the comment section of my last video constantly writing me, he's debunking my videos. He hasn't debunked one of my videos. You have to review a video to debunk it. You can't make...
Well, I am doing it right now, and not only have I reviewed a lot of your videos, because if you go to here, you can see that I am debunking this video on live, and in the other video, look, you can also see that I am debunking these videos on live, okay? But also, if you go right here, I am debunking the video that you're talking about, and what your standard for a debunk is completely arbitrary. When I am able to destroy all of your arguments with just comments and without needing a complete review of the video that you are posting, okay, you cannot just say you have to review it, because the comments have already achieved the purpose of debunking what you're saying. You are just changing the topic because you can respond to the comments.
And fabricate stuff, you understand me, in your videos, saying that
No, I haven't fabricated any of that stuff. It's literally backed up by a lot of studies. As a matter of fact, 41 pages of studies debunking you. You're completely dumb and delusional.
said this. Meanwhile, you didn't prove I said that because you fabricated.
Yes, I proved it. I put your videos in my reviews right here. You can see it.
The strings, which is the central nervous system, mechanical tension is not.
He says that muscle damage is not the driver. What mechanical tension is. What mechanical tension is muscle damage.
Mechanical tension is not.
As you have already seen, I am putting your videos, I am letting you talk, and then I am debunking you. So I am doing the thing that you are claiming that I don't do.
Fabricated that. I never did say that. I never said mechanical tension was never muscle damage in my video. I said that that idiot lifter said that muscle damage wasn't the driver. Said mechanical
What
tension
are
was
you
never
even
muscle
saying?
damage in my.
It's a complete strawman. I didn't even talk about it. Let's repeat it. I never said mechanical tension was never muscle damage. At which point did I say that it was never muscle damage? I said that mechanical tension is not always accompanied by damage, and it is a driver of hypertrophy. Well, it's completely explained in that video. Completely by its own. You don't need this byproduct for mechanical tension to activate muscle damage, providing that some mechanisms work exclusively by mechanical tension, like sarcoplasmic mechanosensors and solute gradients, concentration gradients. All of that only work by mechanical tension, okay? They do not need this damage, this byproduct, at any point in this of their stages of their own mechanisms, okay? They do not need this byproduct of mechanical tension at any point in the mechanism that works to activate the satellite cells and make them donate their nuclei and produce growth. Damage is not involved at any point in these things that I am describing, okay? So you cannot blame it onto damage. That is not only begging the question, but also a baseless assertion. Provided that at any point, at no point, they are talking about the damage produced by mechanical tension. They are just talking about mechanical tension. You cannot make things out of thin air. Just because something is related to the other thing, as one being a byproduct of the other, you cannot say that they are referring to the byproduct, okay? You're making that out of thin air.
My video, I said that that idiot lifter said that muscle damage wasn't the driver of hypertrophy or growth.
Because it isn't.
Whatever. He said that mechanical tension was, and I said mechanical tension is muscle damage.
And I debunked that notion. You completely misunderstood what I said in my video. You're really dumb.
I say it in my video, and I go, what the fuck is he talking about? Because everything is damage. Look, let's explain this.
No, everything is not damage. Muscle tension can cause damage, but it doesn't mean that it works by the damage that it causes. And I have already provided all of these mechanisms that work with mechanical tension alone. And they don't watch out for damage. Osmotic sensors, gradient concentration, cell swelling, this mechanism right here, which works by muscle stretch or injury. It is not talking about exercise injury induced by muscle stretch or injury, because it would only say injury if it was talking about that, because injury already encompasses the both of those things, so it is differentiating them and talking about them as a causal factor all alone, all of their own. So you have also this mechanism, which acts by the release of NO. You have the IGF-1 factor study right here, the Schoenfeld study that also proves that, the Fukada study, the study about repeated bounce effect. You have a complete debunking of every single point about muscle damage being the only driver of hypertrophy right here. That's completely dumb.
One more time in the video, okay? Everything that you're doing in a fucking gym is damage. You're damaging.
No, everything that you do in a gym is not damage. Suppose that you are doing jumping jacks, that's not damage.
Some tissue.
That's aerobic training.
I don't know. It could be through hypoxia, ROS. It could be micro injuries. Fuck. It could.
Yes, you are going to get damage no matter what, but damage is not the thing controlling growth, because if it were controlling growth, less damage should imply less growth, and that is not at all what we see right here, and also not at all what we see when you consider that they are mechanisms that work strictly by muscle tension alone. They don't involve damage at any point in their processes, okay?
It could be anything, you understand me? It could be anything. It could be disease. I don't know. Maybe you have a disease. You're diseased, injuring it through disease. I don't know what the fuck people are doing in the gym these days, get it? They're crazy. You understand me? So there are many different kinds of injuries to muscle fibers, to skeletal muscle, to skeletal, whatever, even your skin. Like I said.
Yes, and any kind of thing that compromises structural Sorry, I completely forgot what I was going to say. Yes. And every injury that changes the structure of the cells or that compromises it, okay, is going to cause your cells to release some factors that cause satellite cell activation. As a matter of fact, ROS works in the same way in which micro injuries work. They both release mitogens because they both work by stressing the overall structure of the myofibers and the extracellular matrix. So if they both work by the same basic principles, you cannot just make an arbitrary distinction between both. They work in the exact same way.
There are different
They damage the structure of the myofibers, and therefore they both cause the release of mitogens. Because for mitogens to be released, you need damage to the structure of myofibers. So if something causes damage to the structure of myofibers, it doesn't matter if it is ROS, if it's hypoxia, or if it is cell swelling. Well, in this case, cell swelling primarily works by compromising the structure of the muscle fibers, not by strictly damaging it and breaking it. But if anything of that causes some kind of compromission to the structure of the myofiber, then you are going to have mitogens released and you are going to have growth. That's all. You don't have to make this arbitrary distinction to be convenient for your arguments, okay?
There's many different types of injuries that could happen. It could be toxicity. You could be filled with oxalates and lifting weights, and these oxalates, they form crystals. They could be damaging your skeletal muscle and your brain.
I imagine that he thinks as oxalate as crystals, like this right here. Even though oxalate crystals don't have anything to do with the crystals you're watching right here. They have more to do with this right here. This right here. A crystalline structure of the atom, okay? That's completely dumb. You don't even know what you're talking about.
Brain cells, dude. The guy that keeps writing me. Your brain and your eyeballs. You understand me? And maybe your pee-pee, your dick is not working too because of those crystals flowing through when you're peeing. That's why you keep writing crazy things about you debunking my videos and stuff, whatever.
Sorry for giving you criticism that completely dismembers everything you say. It shows that you cannot deal with people thinking the opposite that you think. And especially, it's quite hypocritical considering that you try to critique every single content creator you find on YouTube. So you cannot be crying because someone is responding to you.
A bot and shit and everything. Get it, broski? So yeah, so we know that toxicity, the environment, what you're eating can cause tissue damage. And this can accelerate it when you go to the gym. And we know that when you lift weights, you're accelerated aging because you're damaging. If you're damaging, you're accelerated aging.
No, if you're damaging, you're not necessarily accelerating aging because the adaptations you get in return, you have always to think about the cost-benefit analysis, okay? If God suddenly offers you 100 years more of life, by breaking your leg, then you are going to obviously accept that offer because you think about the reward, not about the process that you have to go through, okay? And exercise works in this exact same way. You have to produce mechanical stress. You have to damage yourself in order to get adaptations, okay, that make the damage that you receive on your daily life by just existing, yes, red meat, causes oxidative damage. Anything you eat causes oxidative damage because it's a byproduct of metabolic processes. So each time you digest anything, you cause oxidative damage. So these adaptations that you get from exercise, which are decreased oxidative stress, you can watch it right here. I think that I have a video about oxidative stress and exercise, which is this one right here about nutrition facts. It's literally the first one that appears right here, in which they talk about runners, and Also, you have sorry, this wasn't the video that I was talking about. This one right here, Doc on the Run. They strictly talk about runners and how oxidative stress really increases overall in people who train, making you live longer. Okay.
chronically damaging. You're not regenerating. You're damaging, and if anything, you'll be losing.
Oh, so you're now saying that I am getting more damage than you, even though I have the repeated bout effect that you constantly criticize acting upon myself. You are not consistent in even two sentences that follow one to the other. It's completely dumb what's happening right here. It is unbelievable, really.
You're getting smaller because you're not allowing your body to repair, and then you fuck the regenerative process and you shrink. Go see House of Hypertrophy. He just posted a new video. Maybe you'll learn something.
Yes, House of Hypertrophy debunks what you say. He has a video talking about why muscle damage is not the thing that promotes growth. You have the article right here. Let's see. Well, let's see where it is. The biggest muscle building myth. You have it right here. They talk about damage being the primary stimulus for gains, and they debunk it. Well, this is not the complete version. You have to search for House of Hypertrophy. Damage. Okay, you have to go to their website right here, which I have already gone into. And they debunk what you say about micro tears being the only way in which you can grow. It says that in the title, micro tears do not stimulate hypertrophy. Okay, so it debunks you.
Watching it. It's brand new. I don't know. Fuck.
And also,
You're not
let's
regenerating.
review the stupidity that he has said right here.
You know that toxicity, the environment, what you're eating can cause tissue damage, and this can accelerate it when you go to the gym. And we know that when you lift weights, you're accelerated aging because you're damaging. If you're damaging, you're
Well, that was already debunked.
accelerated aging. You're
That's debunked.
chronically damaging. You're not regenerating. You're damaging, and if anything,
Also debunked based on the description that he has of the repeated bout effect. This thing right here is the one that I want to address.
you'll be losing. You're getting smaller because you're not allowing your body to repair, and then you fuck the regenerative process and you shrink. Go
In which way have we fucked the regenerative process, considering that we have the repeated bout effect acting upon us and making the regeneration process happen faster, and having to deal with less injuries so that there is more protein available for growing instead for repairing? Because yeah, when you have the repeated bout effect, you have to spend less resources repairing your muscles. You have more resources for just making them bigger. That's also common sense.
see House of Hypertrophy. He just posted a new video. Maybe you'll learn something
And he also debunked what you say in this article right here.
watching it. It's brand new. I don't know. Maybe you should go check it out. I don't know. Because people are just too stupid to understand anything out there. Here, maybe I'll show it to you. It looks like this. Yeah, it looks like this. Right there. There it is. This is working. 90% still believe this builds more muscle. 90% of people, whatever. Actually, that's probably one of his best videos, I'd say. It's pretty close. Yeah, it's not bad, actually. So there you go, man. That's the driver of hypertrophy, my friends. I don't know what else to tell you. I'll see you in the next video. Tell me what you think about that. Like, subscribe. Protein synthesis.
Same thing I'm going to say you. Like and subscribe. Tell me in the comments if you want me to keep debunking what he's saying. This is not the part 2, the continuation of the part 2 of my videos. This is a video I decided to do now in response to what he was saying. That's something that I wanted to do just because, let's say. So it's not going to have a how can I say it, a thumbnail nor anything like that, because it's an improvised video, okay?